Our immune system continuously protects us from any pathogen that is trying to invade our system. Now the process involves majorly recognizing between self and the non-self. But how does a cell communicate with the immune system to induce an immune response or not? Now this is done by a group of surface proteins which are called MHC proteins or major histocompatibility complex proteins. Now the gene of MHC is in the genome of every jawed vertebrates. These are the polymorphic genes linked together to code for a surface protein essential for adaptive immunity in a person. The locus got its name because it was discovered during the study of tissue compatibility upon transplantation. In a cell, protein molecules of the host's own phenotype or other biological entities are continually synthesized and degraded. Each MHC molecule on the cell surface displays a small peptide molecular fraction of protein called epitope. The presented self-antigen prevents an organism's immune system from targeting its own cells. Presentation of pathogen-derived proteins result in the elimination of infected cells by the immune system. Three scientists genetically determined the structure of the cell surface that regulates immune response. George D. Snell, Jean, and Porge. Now the gene is presented in all jawed vertebrates, and the MHC molecule is also called HLA or human leukocyte antigen complex in humans. MHC gene family is divided into three subgroups: MHC class 1, 2, and 3. Now the class 1 encodes for protein binding and molecule antigen processing, and the expression is one gene, that is alpha, whose ligands are CD8 receptor, which are recognized in cytotoxic T cells, and inhibitory receptors bound by NK cells. Second class molecules also have a peptide binding domain and protein assisting for antigen loading into MHC's second peptide binding domain. Now the two chains, that is alpha and beta, whose ligand are CD4 receptor, which are bound by T helper cells. The third group is a group of proteins which perform various immunological functions like other than antigen processing and presentation, such as components of complement cascade. Now here is simplified organization of major histocompatibility complex, that is MHC, in mouse and human. Now MHC is referred to as H2 complex in mice, whereas HLA complex in human. In both species, MHC is organized into a number of regions of encoding class 1, class 2, class 3 gene products. Here we have labeled the class 1 product with pink color, class 2 with blue, and class 3 with green. The class 3 products include complement, that is C complement proteins, and the tumor necrosis factor, that is TNF beta and TNF alpha, as you can see in this figure here. MHC class 1. MHC class 1 gene encodes glycoprotein molecules which expressed on the surface of all nucleated cells and platelets. MHC1 molecule contains a 45 kilodalton alpha chain associated non-covalently with 12 kilodalton beta 2 microglobulin molecule. Association of alpha chain and beta 2 microglobulin is required for expression of class 1 MHC molecule on cell membrane. Alpha chain of MHC1. The alpha chain is a transmembrane glycoprotein encoded by polymorphic gene within a B, and C region of human HLA complex. The alpha chain is anchored in a plasma membrane by its hydrophobic transmembrane segment and hydrophilic cytoplasmic tail. The alpha chain is made up of three domains, alpha 1, 2, and 3, each domain containing approximately 90 amino acids, a transmembrane domain of 25 hydrophobic amino acids followed by a short stretch of charged hydrophilic amino acids of cytoplasmic tail of 30 amino acids. Alpha 1 and 2 domain interact to form a deep groove on the top of which a peptide binding cleft is there. It can bind to antigen of 8 to 10 amino acids long. Alpha 3 and beta 2 are organized into beta-plated sheets, each formed by antiparallel beta strands of amino acids. This structure is known as immunoglobulin fold. Because of this structure, alpha chain and beta 2 microglobulin are classified as a member of immunoglobulin superfamily receptor. Beta 2 microglobulin of MHC1. Beta 2 microglobulin is a protein encoded by highly conserved gene located on different chromosomes. Beta 2 microglobulin is similar in size and organization to alpha 3 domain. Beta 2 microglobulin does not contain transmembrane region and is non-covalently linked with alpha chain. Now here is the structure for MHC1 molecule. As we can see, it contains 4 parts: alpha 1 domain, alpha 2 domain, 3 domain, and beta 2 microglobulin. And as described, there is a protein binding cleft between alpha 1 and alpha 2 domain. And as we can see in the diagram B, the alpha 1 and alpha 2 domain are interconnected by beta-plated sheets. Major function of MHC1 is to bind peptide antigens and to present to CD8 receptor on T helper cells. CD8 T cells are specific for MHC1 antigen. MHC1 binds to endogenous antigen and present to the T helper cells. MHC1 molecules are found on the surface of all nucleated cells. MHC class 2. MHC class 2 is a glycoprotein molecule expressed primarily on antigen-presenting cells such as macrophages, dendritic cells, and B cells. MHC class 2 molecule contains two different polypeptide chains, one 33 kilodalton alpha chain and second is 28 kilodalton beta chain, which are associated by non-covalent interactions. Alpha chain and beta chain of MHC2 is a membrane-bounded glycoprotein that contains external domain, a transmembrane segment of a cytoplasmic tail. Alpha 1 chain and beta chain are made up of two domains, alpha 1 and alpha 2, and beta 1 and beta 2 respectively. Peptide binding cleft is open-ended groove formed between alpha and beta chain at proximal end. The cleft can bind antigenic peptide of 13 to 18 amino acid long. Now function of MHC class 2. The major function of MHC class 2 molecule is to bind peptide antigen and present to CD4 T cells. The MHC2 are found on the surface of antigen presenting cells APCS. The CD4 T cells are specific for MHC2. It activates beta cells for antibody production. MHC2 plays a significant role in graft versus host response and is in mixed lymphocyte reaction (MLR) because the immune response gene is identical to MHC2 in human. Here is the structure for MHC class 2 molecules. MHC3. MHC3 are diverse group of molecules that serves as a wide variety of functions in immune system. MHC3 are not marker on cell surface. MHC class 3 genes are located on chromosome 6 at the location 6p21.3. In human, it covers 700 kilobase pair and contains 61 genes. function of MHC class 3. It involves an inflammation caused by cytokines, tumors, and necrosis factors, etc. Other immune response other than antigen processing and presentation such as complement cascades. Thank you.